Morning, good evening, everybody, wherever you might be. I am Rob Burgess, and I head up business development for Sino-Biological. And I would like to officially welcome everyone to the next installment in the Sino-Biological sponsored and hosted seminar series called BioTalk Tuesdays, where we hear from leading researchers worldwide on cutting-edge technologies and about their research, and also about some real-world applications of those technologies and advancements. In just a second, I'm going to introduce to you a wonderful speaker. But before I do that, I just want to emphasize one housekeeping issue, and that is that we recommend, if you have a question for the speaker, that you simply type it into the chat box. And at the end of the actual meeting, we will run through all of the chat box questions, and I will ask them verbally of our speaker. And then we'll try to get as many of your questions answered as possible. Also, please shout out in the chat box and say hello and say what country or part of the world you're from. That's always very interesting for us as well, to see where our attendees are logging in from. So with that, and without further ado, we'll go ahead and start. And it's my pleasure and honor to introduce to everyone our distinguished speaker today. It's Dr. Yu Zhang. Dr. Zhang is an assistant professor in microbiology and molecular genetics at Michigan State University. Dr. Zhang obtained her MD degree at Capital Medical University in China and received her doctorate at Georgia Tech in Atlanta. It was here that she identified key genetic players in preventing genome structure variations mediated by unstable DNA repeats. She further then completed her postdoctoral studies under Dr. Frederick Alt at Boston Children's Hospital and Harvard Medical School. And it was here where she also revealed a fundamental role of chromatin loop extrusion in mediating VDJ recombination, specifically in B-cell development. So Dr. Zhang's lab now at Michigan State University is focused on identifying key mechanisms that really promote antibody diversification, but also suppress associated oncogenic lesions in B lymphocytes. And so with that, the title of Dr. Zhang's talk is The Role of Chromatin Loop Extrusion in Antibody Diversification. Dr. Zhang, welcome. It's a pleasure to have you today, and I will now turn the screen over to you so you can display your slides.
Sure. Thanks, Rob. Okay. Thanks to Rob to the very kind introduction. And one sec. Today, I'm going to present our recent advances in understanding the role of chromatin loop extrusion in antibody diversification. Oh, here. So let me arrange my panels here a little bit better to show my screen. Can everybody see my slides? Can you hear me?
Yeah, we can see it. Looks good, Dr. Zhang.
Okay, great. Yeah. So the numerous antibodies in our body is generated through a process called antibody diversification that plays a major role in protecting us from infection, diseases, and various cancers. And conversely, defects in this process can underlie various diseases ranging from immunodeficiency and autoimmune diseases, and also can contribute to the oncogenic translocations reported in leukemia and lymphomas. Therefore, understanding antibody diversification is really important to understand these diseases, and also can guide in the therapeutic drug design. And this provides a drive for us to study this process. So to generate these antibodies, there are three major processes. And before that, let's talk a little bit about antibody, which is comprising of like 2 heavy chains and 2 light chains. And each type of chain also it consists of a variable region that bind to the antigens and a constant region that direct the antigenic stimulation into proper immune response. So the 3 process of antibody diversification occurs in the B lymphocytes. And the first one is called mediate recombination that assembles the numerous variable exons in early B cells. And upon antigen activation in mature B cells, there is another process called somatic hypermutation that further introduces antibody diversification through introducing point mutations. And this allows the selection of high affinity expression B cells. And a third process is called class-switch recombination that also occurs in mature B cells upon activation. And they change the isotopes or classes of the heavy chains and therefore produce the proper effector functions for each antigenic challenge. And okay, let me go to the next slide. So, and this talk will majorly focus on mediate recombination. So mediate recombination is a process that cut and paste different VDJ segments on the antigen receptor node side. And those segments can be spread up to megabase distance in chromatin. And in both human and the mouse, there are 7 antigen receptor node sites. 3 of them, including the immunoglobulin heavy chain locus, they undergo ordered mediate recombination that has a D2J join first, followed by V2DJ join. And the 4 other antigen receptor node sites do not have these segments and then undergoes direct V2J join. So since the discovery of mediate recombination in 1970s, many seminal findings have been discovered toward understanding of this signature activity of antibody diversification. So this includes the identification of the RAG enzyme that initiate the cutting step and the identification of non-homogeneous end joining process that finish the pasting process, and also the emphasis of chromatin accessibility as important regulation means to control mediate recombination. However, in spite of this progress, there is a key question remaining regarding the control of mediate recombination. That is, how does the RAG protein find their targets at the first place, given the complexity of the genome and also the size of this antigen receptor node site? And the challenge to this question majorly lies on the inherent complexity of the antigen receptor node sites themselves compared with other genes in the genome, and also lack of sensitive assays that can track the changes in the antigen receptor node site. And thankfully to the recent advances in the next generation sequencing technologies and also the genome engineering technologies, we are able to address this question now. And recent years have revealed novel mechanisms driving RAG targeting, both in the normal mediate recombinations for antibody diversification and also abnormal mediate recombination that leads to oncogenic translocations. So just a brief review of mediate recombination. So mediate recombination occurs between a pair of substrates called recombination signal sequence, also called RSS. And the RAG initiate this process by targeting to the RSS and cleaves between at the border between the RSS and its flanking coding sequences. This generates the blunt-ended signal ends and helping capped coding ends. And then a non-homogeneous end joining repair pathway repairs these breaks. And this is directed in such a way that coding end joins to coding end and a signal end joins to signal end. Where the signal ends, the blunt ends can be fused perfectly by XRCC4 and ligase 4. For the coding end, the helpings has to be first opened or processed by the DNA PKs and the ultimers, and then further it can be diversified by TDT by adding random nucleotides before the ligation. And of course, there are other DNA repair proteins that cooperate in this process to complete the recombination. And for the RAG endonuclease, it is a Y-structured endonuclease and comprised of, it is a heterotetramer that is comprised of 2 RAG1 and 2 RAG2. So RAG1 is a kinetic subunit, and RAG2 is a very important regulatory cofactor that is important for both the cutting activity of RAG1 and also important for its DNA binding activity. And the RAG substrates ORS is consist of conserved haptomer and a nonomer that are separated by either a 12 base pair spacer or 23 base pair spacer. An important note here is that the first nucleotide of a haptomer, also CAC motif, is very important for the RAG cleavage activity. So mutation of this nucleotide will lead to cleavage of RAG. And it's also important to note that RAG functions under 1223 rule, which means that efficient video recombination only occurs between a pair of 12 RSS and 23 RSS. So although the major outcomes of video recombination is the proper assembly of the antigen receptor nuclei, a minor fraction can go away. So the double-strand breaks from multiple runs of video recombination can also join to RAG-independent breaks from other sources, such as oxidative damage or some other enzymatic breaks in the genome. And this can lead to oncogenic translocations that have a very strong enhancement in antigen receptor nuclei placed next to oncogenes, and therefore need to multiple kinds of informers. On the other hand, RAG can also target to Crispr substrates in the genome outside of antigen receptor nuclei. They are called cryptic RSS, and this can lead to recurrent interstitial deletions and other genome structural variations that are often reported in acute lymphoblastic leukemias. It's also important to note that computational analysis predicted an average density of 1 cryptic RSS per 500 base pair in the human genome. So without proper regulation, the RAG enzyme can tear the genome in seconds. So here comes to the first question, how is video recombination controlled to minimize collateral damage? To address this question, it's actually very important to have a very sensitive assay that can detect the RAG activity, which includes both on-target activity and off-target activity. And so we took advantage of the high-throughput genome-wide translocation sequencing, also called HGTS. So HGTS use defined beta double-strand break to capture unknown genome-wide double-strand breaks that joins to this base break. And this base break can be ectopically introduced by designer nuclease such as IC1 or Cas9, or it can be endogenous breaks such as RAG and AID. So with this, this is an example showing that a base break introduced by IC1 were used to successfully detect the genome-wide breaks caused by AID, denoted in red here, and also the IC1 off-target is denoted by blue here. And this assay was further improved greatly in its resolution and sensitivity by adding a step of linear amplification mediated PCR, which turned it to so-called non-matched GTS. So with this, when we place a bait primer next to a signal end or a code end, a rag break, we're able to capture all of the breaks genome-wide that join into this specific rag break. This include breaks of other sources or breaks at cryptic RSS. And of course, also all of the on-targets. So with this technology, we were able to detect thousands of cryptic RSS in a mouse recurrent multiple B-cell lymphoma model. This was really unexpected at that time because very few cryptic RSS were reported in the literature. So in this model, a DG beta cassette from T-cell beta loci were introduced into the first intron of the CMYC gene on chromosome 15. And a bait primer of non-matched GTS was placed next to the D segments here to detect all of the breaks joining to this D RSS. And what we found is that majority of the breaks were within chromosome 15, and a zoom in of this region showed that, interestingly, they look like to be confined within a 1.5 megabase region near the CMYC. So we did a further zoom in of this region, and this shows the illustration of this region, and CMYC is here. And here is the HGTS profile for the rag targeting activity. So the beta is here, and then the red peaks bars shows the breaks that are joining initially to the bait break, and the blue lines here shows the breaks joining immersionally to the bait break. So first impression you got from here would be that actually the initial joining is predominant compared with immersional joining, right? So then we further found that the boundary of the breaks localize very well with the CTCF and RAG21 ChIP-seq signal. And further on, we found that due to based on published chromatin interaction assay, we found this domain, this region is a domain that has increased interaction within the domain itself. And it also has a subdomain, which is around 800 kb. And this also correlates with increased intensive breaks within this region. And actually the breaks ends sharply at the boundaries of this domain. So this phenomenon is not confined to this specific loci. We tested 12 other loci genome-wide and find similar findings. So overall, this study revealed 3 important features of rag off-target activity. So first of all, it's obvious that bona fide RSS, we tested actually both ectopically inserted and also endogenous one from the IgH locus. So we know that bona fide RSS, they can activate thousands of cryptic RSS. We know they are cryptic RSS, or in other words, rag substrates, because they have 2 important features. One is that they all contain the CAC motif, which I just mentioned earlier, that is critical for rag cleavage. And second, the activity or occurrence completely depend on the rag presence. And second, what we found is that there is a strong bias toward joining of convergent creepers that leads to deletional joining. So in this illustration, what you can see here is that the yellow one is represents the bona fide RSS, and the red triangles denotes the creepy RSS convergent to the beta RSS, and then gray ones denotes creepy RSS in the same orientation with beta RSS. So more than 95% of junction we found occurred as the creepy RSS that are convergent to the beta RSS. So the third feature is that this RAG activity or creepy RSS target activity is largely confined within the loop domains. So this observation together led us to propose a RAG scanning model. So in this model, RAG scans on chromatin up to megabase distance until it's arrested by the boundary of the chromatin loop domains. So this, the feature of this model is that it's a linear process. Therefore, it will only see convergent substrates during this process, but the substrates of other orientations cannot be seen. And this is in contrary to the traditional diffusional access model, in which the orientation of the RSS should not matter, because, I mean, using Brownian motion, you should be able to access CAC or creepy RSS on either orientation. And of course, it's worth to note that the frequency of this convergent or RSS of the same orientation is very similar on the genome. This strong bias is not due to like excessive presence of one orientation of CACs versus the other. Okay, so then the next question we asked, oh, there's one thing I have to note that. So this actually RAG scanning activity help us to provide a mechanistic basis for the recurrent interstitial deletions found in many leukemia and lymphomas. So the next question we ask is, what is the underlying mechanisms of RAG scanning? An important concept we just mentioned is loop domains. So based on the chromatin interaction assays, especially HASIs, it was found that eukaryotic genomes are organized into megabase or sub-megabase topologically associated domains, also called TADs. And it was further found that these TADs, many of them, they are contact domains that have these endpoints with pronounced interactions and therefore generate a loop. And they are called chromatin loop domains. And they have been found to be structural and functional units of the eukaryotic genomes and being implicated in many important biological activities, including replication, transcription, gene repair. So also important feature of these loop domains that they're often co-anchored by CTCF and Cohesing. And CTCF bound to CTCF-binding elements, also called CBEs. CBEs have orientations. So the two CBEs can either be convergent to each other, or same orientation to each other, or divergent to each other. So importantly, the loop anchors of the loop domains are usually associated with pairs of CBEs that are convergent with each other. So another important factor here is cohesing, which is a ring structure that has PT-pace activity. Based on this observation together, a loop extrusion model was proposed for chromatin loop domain formation. And in this model, cohesin loaded onto chromatin can progressively extrude chromatin into larger and larger loops until they're arrested by CTCF that are bound to convergent CBEs. And notably, since this is also a linear process, it does not see CTCF of the same orientation, but only sees CTCF of convergent orientation. And this model has been supported by many evidence in recent years. So if you look at RAG scanning and loop extrusion, it's not difficult to find out that they actually share striking similarities. So they are both considered as linear process. They both employ convergent rule, and they both are restricted by loop anchors. Since RAG protein itself does not have ATPase activity, RAG scanning has to be a passive process that is driven by some mechanisms. Therefore, these features together strongly suggest that loop extrusion is the driving force for RAG scanning. So we test this hypothesis at the mouse immunoglobulin heavy chain locus. So the HH locus have a very unique organization. At the 3' domain of this locus, it is the DJ domain that has all of the D's and the J segments here. And this domain is anchored upstream by the CB-based regulator element called HCl1, and downstream by a cluster of 10 CBEs called 3' CBEs. And in this domain, there is a recombination center that is consisted of all of the J's, the intronic enhancers, and the last D's. And they are very rich for transcription activity and recombinding, and it serves as a hub to capture other partner process for mediate recombination. And upstream of the HCl1, there is hundreds of these that spread it over a very large 2.4 megabase distance. And there are also multiple CBEs within this locus. In the proximal V's, there are CBEs associated with each RSS, and in the distal V's, CBEs is more dispersed within the region. So because the G segments are flanked by RSS on both sides, they can join to both J RSS and also V23 RSS. And HH-mediated recombination is an older process that I mentioned earlier. And in that V2J joins first, followed by V2DJ join. So we first looked at the off-target activity of this DJ domain. And we found actually interestingly, we found actually another interesting feature of RAG scanning, which is that the scanning directionality is determined by RSS orientation. So here only shows the off targets, only shows the bait RSS from DQ52 and also the off targets of this region, because we are focused on off target activity here now. Because the DQ52 is flanked by RSS on both sides, we bait either from upstream RSS or downstream RSS to look for the RAG scanning activity. And interestingly, the upstream RSS only initiate RAG scanning activity in the upstream region, and is arrested at the HCl1, which is a CB-based regulator. And the downstream RSS only initiates RAG scanning in the downstream region, and is also arrested by a CBE block. Therefore, RAG scanning has a directionality. Also, notably, the CBE barriers are hotspot for RAG off-target activity, means they are accumulating RAG off-targets here. And also, beyond CBEs, we found that the S-gamma 2B region in this downstream scanning paths also have strong RAG activity accumulated here. And this is most likely because it's a very robust transcriptional sites. And we will go back to this phenomenon later on. So to test the role of loop extrusion in RAG scanning, we decided to employ a roadblock and to see whether a roadblock can impede RAG scanning via impeding loop extrusion. And to do this, we used the nucleus delta Cas9 as such a roadblock. And because it does not have DNA cleavage activity, but retains, because it doesn't have the cleavage activity, but retains its DNA binding activity. And also we took advantage of the S-gamma-1 region at the downstream scanning pathway of the DJ domain. This is because this S-gamma-1 region is very repetitive, and it can provide multiple Cas9 binding sites, and therefore could presumably form a very strong barrier. So what we can see here is in a control string, there is a strong downstream scanning activity and an accumulation of the RAG activity at the 3' base and S-gamma-2b region. And upon introduction of the Cas9 blockade, what we can see is significantly decreased scan activity at the region past the block, but instead there is accumulation of the RAG activity at the S-gamma-1 region. So the steric hindrance imposed by the Cas9 indeed can impede RAG scanning and accumulate excessive RAG activity at the block region. So we next test the effect of this block on loop extrusion. And for this, we employed the 3C-seq, which use the bait, the recombination center as a bait site to detect its genome-wide interaction partner. And what we can see here is that in the control string, there is a strong interaction of this beta site with the S-gamma-2b region and 3' CBEs region. However, upon introduction of the Cas9 blockade, there actually appears a new interaction peak around the blockade site at S-gamma-1, but correspondingly, there is significant decrease of interaction at the downstream regions. And the appearance of this block, interaction peak in the Cas9 blockade string is not due to a transcription increase, indicating it's a transcription independent mechanism. And also importantly, there appears a new cohesin-binding peak at the Cas9 blockade site. So this data together shows you that the steric hindrance imposed by the Cas9 significantly impedes the cohesin-mediated loop extrusion via formation of new loop with a blockade site. And we further show that this is not specific to that Cas9 blockade, as the active transcription at S-gamma-2b region can similarly impede RAG scanning. So in the white string, as you can see, strong transcription activity is across this region, which correspond to strong interaction peaks and also strong RAG activity. Upon deletion of the promoter of this region, it eliminates the transcription here, which eliminates its interaction peak with the recombination center, and also eliminates the RAG activity here. And instead, RAG scanning now can pass this region more smoothly Therefore, there is an increased downstream reg scanning activity at the 3' CBEs. So taken together, this evidence shows that loop fusion indeed promotes reg scanning, and because the target and endogenous pockets can impede reg scanning via impeding loop progression. Also, an important note here is that various loop fusion obstacles, including CTCF sites, like the AGCL1 3' CBAs, active transcription, like the ones seen at the S-gamma-2B region, and other protein complex binding, like that Cas9, they can all promote enhanced mediate recombination activity at the impeded regions due to the pronounced local chromatin interactions, as shown by the 3C-seq. And this can help to explain why reg targets at certain reg targets are much more highly used compared with others. And also, paper later on provide further evidence of this model, because it showed that removal of cohesin abolished IgH chromatin looping and the mediate recombination. So the next question we asked is, how is reg targeting activity regulated at antigen receptor loci? Is reg scanning specific for off targets, or it also has a physiologic importance? So for this, we also used the mouse IgH locus as a model system. As we just introduced, at IgH locus, the first step of mediate recombination is a D2J join. And for this, actually D2J join is featured by a very strong bias toward deletional joining. Okay, deletional is kind of appears again and again here. So what does that mean? If you look at the DJ domain, there are multiple properties here, but for all of them, the deletional joining is significantly higher than inversional joining, and the frequency can be as different as 1,000-fold. Although if you look at the D segment itself that is flanked by 2 RSS, both deletional joining caused by the downstream convergent RSS with J's, or inversional joining caused by upstream RSS that is in the same orientation as J's, they should theoretically occur the same difference because they both follow the 12-23 rule. So this phenomenon was non-attributed to the difference in the strength of these 2 RSS that can recombine to J RSS. But as we introduced reg scanning and provide a lot of evidence for reg scanning in off targets, it's just natural to think that this can also be be driven by reg scanning process. So to test these 2 possible mechanisms, we did directly by targeted mutagenesis. So for this, we first use the very highly used DFS 6.1. As you can see in the wild type situation, the deletional joining using the downstream RSS is much higher than the inversional joining using the upstream RSS. So we first inverted the downstream DRSS, and this abolished its convergency with the JRSS. So what you can see here is that it's also almost abolished its activity, recombination activity with the JRSS, and renders it as inert as this upstream DRSS. So we next inverted this upstream DRSS, and now both RSS are convergent with the GRSS. So what you can see is there is a dramatic increase or gain of recombination activity of this upstream DRSS now is that is convergent with the GRSS. And also the two RSS activity almost comparable. We then further inverted the whole cassette, which reversed the orientation of the two RSS linking the Ds, and this also reversed its potential to join into the GRSS, because now the downstream DRSS predominantly joins to the Js, but upstream DRSS almost has no recombination activity with the GRSS. So this data clearly shows that DRSS orientation, rather than a sequence, RSS sequence feature, determines the deletional D2J recombination, and strongly support that RAC scanning promotes physiologic T2J joining. So how about V2DJ joining process? So this has been nicely tested, or this has been nicely tested by two very elegant studies in 2020 and 2021. So for the V2DJ joining process, a very important process is called edge-edge contraction, because in uncommitted B cells, the V genes are spread over very, very long chromatin distance, and therefore the distal Vs are very far away from the DJ domain. So in order for these distal Vs to have same opportunity to recombine with the DJ domain compared with the proximal ones, this edge contraction process has to occur in the committed B cells that place the distal ones into proximals with the DJ domain. And these two studies shows that there is developmental repression of WAPO protein in the pro-B cells. And a WAPO is a cohesin releaser, or a noder that unloads cohesin from chromatin. Therefore, this physiological depletion of WAPO can increase cohesin chromatin tension time and generates loops with extended size. This needs to enhance loop extrusion and can therefore drives the edge-edge contraction via loop extrusion, and also that RAG long-range scanning to make them reach to the distal Vs to mediate V2DJ recombination. So in summary, what I have shown here is that, first of all, chromatin loop domains is an efficient way to restrict RAG scanning within the loop, therefore prevent spread of RAG activity at antigen receptor nodesi to other genomic nodesi, and therefore prevent unnecessary oncogenic translocations. And also have shown that this RAG scanning have also plays a critical role in promoting properly regulated initial D2J recombination and the V2DJ recombination. And there is a strong evidence supporting that cohesin-mediated loop extrusion is a driving force for RAG scanning. Also, importantly, we need to know that various CBE and non-CBE based mechanisms can be employed to impede loop extrusion and increase local RAG activity, which can potentially increase the utilization of certain V or D segments compared with others. So here I'm going to quickly show you illustration, video to illustrate the process of loop extrusion mediated RAG scanning using D2J recombination as example. So here's the DJ domain that has the Ds and the Js here. And cohesin is noted into the chromatin. It can be noted from various points, and here's showing an example noting right next to the recombination center. And when it starts to extrude, it can present the convergent GRSS to the GRSS to mediate exclusively deletional D2J joining. And this process continues until it hits the IGCL1 boundary. And also this, because DFS1 is very next to the CTCF boundary, it is the most highly utilized DFS1. Okay. So also I wanted to say that beyond vintage recombination, chromatin loop extrusion can also promote class switch recombination, which is another important antibody diversification process that changes the isotopes of the antibodies. And in this Nature paper, it shows that it shows also model of doing this. So I'm going to show you quickly about this model. Sorry, I just realized it's not a full screen anymore. So this model, Cohesing extrusion, mediate the synapsis of a very important enhancer in class switch recombination called 3' regulatory enhancer. And then it mediates the synapse of this enhancer with the donor switch region called switch mu. And then further loop extrusion mediates the synapsis of the acceptor switch region with this recombination center, and also activate the transcription activity of this acceptor region, switch region. And now this forms a full recombination center. And upon AID cleavage, loop extrusion will pull the two break ends into two ends and therefore promote deletional joining, which is a physiologic outcomes of class switch recombination versus the inversional joining. So this video shows you that chromatin loop extrusion can also be the driving force for class switch recombination by participating in the various steps of this process, including the synapses and the breakage and the repair process. So with this, I would like to thank my lab members, my collaborators, and also a funding agency. And I'm going to leave the slides where I'm taking the questions.
Great. Thank you, Dr. Zhang. That's fascinating research you've done on nuclease and DDJ recombination. We've got just a few minutes left, so I'm going to go ahead and jump into the questions. We've got some great questions from the audience. The first question is very disease focused and I believe practical, and I think it's kind of a high level question. And it is from Dr. Ethel Madera. And Ethel asks, how does RAG endonuclease activity and I guess accuracy work in acute cases of smokers, perhaps with chronic thrombosis? So is there a correlation with disease?
You mean specifically to smokers, for example?
Yeah. I mean, is there a decrease in the efficiency or accuracy, I think is what Dr. Madera is asking, for the RAG endonuclease in those who have cells damaged by smoke or have chronic thrombosis?
Oh, I'm not really aware of this kind of studies yet, but if like in old people, there is definitely a decrease in RAG activity, but I'm not sure that's specific to like smokers.
Any other diseases where it might be affected that you know of other than just age?
Yeah, of course, there are a lot of, like, for example, in seeds, there is lack of drug activity that, of course, will lead to immunodeficiency. Yeah.
Mhm.
But because of this loop extrusion model, it's also interesting to know that other than RAG, other regulators of this process can also lead to like immunodeficiency or other diseases. For example, if you have defect in cohesin or its regulators, then it will be highly related to
Yeah, it's not just RAG, it's a lot of the other
yeah.
proteins and molecules involved in the recombination and extrusion, I guess. So,
Yeah.
Razan Khan asks, how could we determine the variable region sequences of an antibody? Is there an in silico platform or a study to accomplish this?
So the human and a mouse genome are all completely sequenced, and it's for specific strains and specific people. So yeah, all of these sequences are available. Yeah.
Yeah, I think it was referring though to a finished processed antibody, like a mature antibody. Is there a way to do that without doing like Edmond degradation sequencing, or how would you go about that? Do you know?
Oh, you mean for the antibody? How do you know the sequence? I think you still have to sequences, do the sequences to know the sequence. Like there is various kinds of sequencing to do the antibody repertoire sequencing. Yeah, especially you need to know both the heavy chain, light chain sequences. So, yeah.
Okay, next question is from my colleague, Amy Sheng at Sino down in the Atlanta area. Go Jackets at Georgia Tech.
Yeah.
Jackets fans on here today. Question is, since RAG scans passively, does it anchor to another protein or does it work freely on the DNA?
Actually, it's a very good question. Since it's passively, and of course, we think it has to be promoted by Cohesing to do the extrusion. But when it before it start the scanning, it has to first bind to one substrates and then look for the other substrates. And what anchors it at that substrates, I think it can be the it can indeed be other proteins bind within that region. For example, transcription factors or other protein, other cohesin or even other RAG proteins. Yeah.
And I have a question from your colleague, Xiang Qiu, says they actively collaborate with you. They're from the NIH. And the question is, do you think IGCR1 still acts as a boundary at DJ recombined IgH allele?
Still act as a boundary and what?
Do you think IGCR1 still acts as a boundary at the DJ recombined IgH allele? So I'm not sure about that question.
Oh, okay, I see. Whether IGCR1 still act as a boundary in the DJ recombined allele. I think it's a very good question because I think the timing of WAPO depletion or the timing of it is not that well known yet. Of course, we based on boosting and study, we know that PAX5 regulates the WAPO depletion, but how quickly it occurs after the DJ recombine is unclear. So in the Abelson cell line, where you do not have this developmental depletion of WAPO, of course, IGC1 still serves as a boundary. But in the developing B cells, at specifically which stage it lost its ability as a boundary because of the enhanced loop extrusion, I think that is an open question and it's a good question. Yeah.
Great. Nadal Kara asked, does this model take into consideration the linkage equilibrium concept, if you're familiar with that?
Sorry?
It's called linkage equilibrium concept. Is that something you're familiar with, or?
Or I'm not familiar with that.
Okay. Okay. Sorry, Nada. We'll have to skip your question. Maybe we can get back to you on that. And Jerry Smith asked the very simple question, does RAG bind to cohesin? I think that's a yes, right?
That is an unknown question. Yeah.
Okay. Good question, Jerry. Eventually somebody will figure that out.
Yeah. It's an important one to figure out. Yeah.
So let's see. Hold on one second. This is a really good question from Cheryl Song, and I was going to ask this too, but what's the clinical application of all this new knowledge for RAG extrusion mechanisms?
What's a clinical application? So that's a good question. I think, for example, for the oncogenic translocation part, we know that we know that interstitial deletions occurs frequently in a lot of cancer, in lot of lymphomas and leukemias, but we don't really know the mechanism before. Now we kind of know that RAG scanning is the mechanical basis for this process, and also for the VDJ repertoire, like whether certain V used more compared with others, is also a non-solved question. And then various findings in these studies can provide insights, like whether it has CTCF sites with next to it, or whether it has like transcriptional activity near the V's. And so how this can be applied to clinical is like, actually, I know that then you can, for example, for HIV vaccines, some models actually employ this knowledge and to try to increase the targeting of specific V's. So to study somatic hypermutation. So I think all of this basic knowledge will find applications.
Yeah, I guarantee you it will. So
yeah.
And Jerry had just typed in, Jerry Smith, I know you didn't get the answer on the cohesin-binding by RAG. I think Dr. John said it's just not known yet. So those studies are forthcoming. It's not known if they directly interact yet. Let's see. Let me just ask you a question if I can. And I'm sure you answered this, Dr. John, but regarding the length of DNA that RAG could really scan and close the loop on, I mean, I know there are, what do you call, some loop domain boundaries, but what's the limit? Is it 2 megabases or 10 or 100? I mean, what's the maximum distance or maybe the maximum size of an extruded loop?
You know, it's actually a very good question. So in a recent study by Fred, so in a B-cell9, when they deplete the WAPO and test how long the RAG scanning can go, or loop extrusion can go, actually, because HH locus is located near telomere, I think it's around maybe 6 to 7 megabase distance near the telomere. What they found is that when they deplete the WAPO, the loop extrusion can go to the very end of telomere. So wow. It can go up to many megabases. But what's the limit? It's difficult to say, and it all depends on, for example, under WAPO depletion, what's the association time of cohesing? What's the maximum association time of cohesing can be on the chromatin, and what's the translocation speed of cohesing? Yeah.
Right. That's fascinating. I used to study methylation and epigenetics, and I remember MECP2 could affect gene transcription over hundreds of megabases. So I know it's not the same as what you're studying, but it's fascinating that these proteins can have such an impact over such long distances of the nucleotide sequences. So one more question, and I think we'll wrap it up. We've had a lot of great questions. I think I'm getting to all of them. I'm just going to read it word for word. It's a little bit long. Since RAG, this is from Kavita Sharma. Since RAG-based scanning is of utmost importance for RSS-based DGH editing, does the recombination pathway get affected if cohesin genes are knocked out by CRISPR or Cas9? So I guess if you remove cohesin, what happens, right?
Yeah, so if removes cohesin, actually it kind of abolished mediate recombination.
Mm-hmm.
So
essential molecule then.
Yeah, it's very essential. Yeah. So, well, in some sense, it abolished mediate recombination that requires a RAG scanning because it's more like because the RAG cannot reach to its targets.
Yeah.
But if in a special case, if the target is very close to its partner, like within, for example, 20 kb, then it can also access to each other via diffusion. So in this case, the activity still remains, but it will abolish most of the mediate recombination across the locus. Yeah.
Thanks for the answer. I was just reminded we're out of time. So I just want to say a few thank yous. First of all, thank you to all the attendees. Very quickly, we had a worldwide attendance, a very diverse audience from India, Germany, Baltimore, Ireland, Boston, of course, Atlanta and the Yellow Jackets, Raleigh-Durham, Uruguay, Nigeria, Toronto, Houston, and Bulgaria. Thank you to all the attendees, and thanks for the great questions. Dr. John, I want to thank you next. That was a wonderful seminar. Fascinating work you've done on the RAG endonuclease and VDJ recombination. Look forward to seeing that in real-world applications as well, as we discussed a bit. Appreciate you attending today and giving us this seminar and lecture. And then finally, once and for all, thank you to Christine Lay, my colleague at Sinobiological, for setting this BioTalk Tuesday webinar up and executing. So with that, we'll say goodbye, good afternoon, and good night to everybody. Thank you.
Thank you, everyone. Bye.